Hello, my name is Hannah Schneider, and I work at the Leibniz Institute for Plant Genetics and Crop Plant Research in Germany, and the institute is also known as the IPK. There, I lead the group Genetics and Physiology of Root Development, and today I'm going to be talking about integrated root cortical tissues for enhanced drought stress tolerance. So not only does root architecture play an important role in soil foraging and soil resource acquisition, but also root anatomy. And that's really what we're focused on is the arrangement of cells and tissues and different cell types inside the root. And you can see, as you can see here, these are a variety of maize and barley root cross sections, and you can really see the diversity in tissues and cell types in these cross sections. Now, one of the major challenges of anatomical phenotyping is imaging. It normally takes a long time and it's very, very laborious. However, in collaboration with Molly Hanlon at the Danforth Center in Missouri and Joel Swift at the University of Kansas, along with Dylan Jones at the IPK, we've developed a root anatomical imaging device that really can speed this process up. So the assembly of this device is relatively simple. We use a USB microscope with UV lighting. We have a stage. In our case, we have an XYZ motorized stage, but that's not necessary. Any maybe single axis stage would do. And access to a 3D printer. And we have 3D designed and 3D printed parts that enable uniform cutting of root cross sections and also the placement of these root segments in front of the camera for rapid imaging. Now, this setup doesn't require the preparation or embedding of tissue in any way. We just take the root, wash it from soil, preserve it in 50% ethanol, take it out and mount it on our cassette for cutting, and we're ready to go. So here's in sort of an image sequence of the cutting steps. So as you see here, you have this is step one, you have the razor blade here, you have a stage here. Here you inlay a cassette with a small groove, and that size of that groove can be different sizes to accommodate different root diameters. You lay your root in there, sweep the razor blade across. Actually, you put a magnetic cover on it to sort of clamp the root in place. Then you sweep the razor blade across, creating a nice, clean cross cutting face that you can image. And so the whole setup looks a little bit like this. Here on top we have the stage controller that controls the XYZ stage, but that component is not necessary. We have a very simple, very cheap USB microscope. That USB microscope has UV lighting. In this case, we also have a supplemental UV ring light source that helps illuminate the sample even more. Here is the 3D printed sample holder where we made that nice cut of the root. We have a motorized XYZ stage. We have a small device to hold the camera, to adjust the camera in the Z direction. And then we have also a 3D printed sample tube holder. So we store our samples in small Eppendorf, 2 milliliter Eppendorf tubes, and then we have a printed QR code label on them that identifies the sample. You can pop that Eppendorf tube in the sample holder, and when you press capture on the software, it not only captures a picture of the root, but it also captures a picture of the QR code, and the software can read the QR code and automatically rename the image file. So this is what an image would look like after it's cut. You can see this is our root here. And we have the scale down here. And then we could also pop in the QR code that identifies the sample if we want. You can see these tiny laterals also got in the picture here. These are just by chance. This is the main root is the big root we're looking at, but you can also capture lateral roots if you're lucky. And here you can see just some more samples of images we can collect. So we have a brace root down in the bottom right corner, in the upper left-hand corner of maize. We also have some crown roots of maize. You can see that we can collect Orencoma lacunae. We can have we see a lateral root emerging from the pericycle here. So we can't capture sort of subcellular structures because the resolution of the camera isn't that great. It's sort of just a quick, easy way to get structural information about the anatomy, orenchyma, sclerenchyma, size and number of xylem vessels. It's a relatively rapid way to capture that information. And an average person can take images, I would say, of between 200 and 400 samples per day. So it's relatively high throughput in terms of root imaging and phenotyping. So one application we use this for is studying the root cortex. The root cortex is highlighted in red here on a maize root cross-section. So the root cortex encompasses everything from including the endodermis all the way up until the epidermis. Epidermis is not included. So from the endodermis to the hypodermis, all that tissue is considered the cortex. It's a primary ground tissue. It's a very large component of the root volume. Early in plant growth, it's primarily comprised of parenchyma, which are very simple, thin-walled living cells. And the cortex really plays an important role in plant growth, structure, and function, and can develop lots of different tissues through different developmental trajectories of these cortical parenchyma cells, which I'll show in the next slide. So some cortical parenchyma cells in some genotypes can develop a tissue called multiserrate cortical sclerenchyma. We abbreviate it as MCS. It's really defined by small cells with very thick cell walls in the outer cortex. So here you can see two maize lines, one with MCS and one without MCS. And we've shown that when you have MCS, you grow better in compacted soils. And here's a bit of a close-up of MCS. So we're really focused on this purple area, this outer part of the root cortex. And in this upper panel here, you can see this is a genotype with no MCS. You can see these are cryo-SEM images. You can see that in those outer cortical tissues, the cell size and cell wall thickness is relatively uniform. While in the lower panel, that would be a genotype with MCS. And you can see just below the epidermis, you have very, very small cells with very, very thick cell walls. And we know that these cell walls are encrusted with lignin, so we've done FTIR spectroscopy. We've done also a lot of histological studies really showing that these cell walls are thickened by the embedding of lignin. And we see a lot of phenotypic variation for MCS in a lot of cereal species, including barley, sorghum, wheat, promillet, maize, like I showed earlier. Barley maybe has the most extreme phenotype of MCS. You can see here that MCS nearly comprises half of the root cortex, which is a very large proportion. Now, we have done a lot of physiological experiments in the greenhouse and in the field showing the utility of MCS under compaction stress tolerance. Now I'm just going to highlight a field experiment here that sort of sums it all up. But we have genotypes with MCS that are represented in blue and without MCS that are represented in red. And in our field study, we had a non-compaction treatment and a compacted treatment. If we just can focus on the compacted treatment here, you can see that genotypes with MCS had a greater D75. And D75 means the depth at which 75% of the root length was at or above. So a greater D75 means your roots went deeper. So when you have MCS in your compacted soils, your roots were able to go deeper. And that resulted in greater dry shoot biomass, presumably because these deeper roots could capture more nutrients and water to sustain more plant growth. So sort of the summary of the story is that when you have MCS, these cell walls are encrusted with lignin that improves the tensile strength and improves the mechanical strength of the root, enabling penetration of hard soils, resulting in greater root depth and greater plant performance under compaction. We also know that MCS is genetically controlled through genome-wide association studies, also through QTL mapping of the IPM population. We've shown we have a significant SNP on chromosome 9 that we're following up with some genome editing to confirm the function of this gene. Now, MCS can have potential utility for a lot of different environments, under a lot of different stresses, but also potential trade-offs, right? So here, if we focus on a genotype with MCS, we can see that from our experiments, we know it has reduced root respiration. From tracing studies, we know that it reduces radial hydraulic conductivity, but also reduces radial water loss. Through also tracing studies with radioactive phosphorus, we've also known that it reduces radial phosphorus and probably other nutrient transport. And it can also act as a mechanical barrier to prevent pathogens and herbivores. So we've done some preliminary studies, for example, with western corn rootworm, and showed that this lignification in outer cortical tissues can really act as a mechanical barrier to prevent root worms from munching on getting in and munching on the cortex. But it can also have potential trade-offs, right? So for example, this mechanical lignin barrier might also prevent AMF colonization, for example. So now I'm going to completely switch gears. We're going to talk about another root trait, another root tissue that develops in the cortex called root cortical senescence. The name of the trait describes exactly what it is. The cortex senesces. This is a common phenomenon in some genotypes of temperate small grains, including wheat, barley, oats. And you can see really it here. On the right-hand side, the cortex is completely almost senesced, leaving the stele. And root cortical senescence progresses, so if we just highlight one root in red here, and we look along the longitudinal gradient of the root, you can see at the root tip, the root cortex is fully intact. And as you move up that longitudinal gradient of the root, you can see the progression of root cortical senescence, eventually only leaving the stele. So in these older tissues, in these older parts of the root, the cortex is senesced in some genotypes. And we've done a lot of greenhouse work, field work, and also simulations with the functional structural plant soil model OpenSimRoot to show some of the benefits of root cortical senescence under low nitrogen, in low nitrogen. So for example, when you have root cortical senescence, that's marked in red bars, no root cortical senescence marked in blue bars. When you have root cortical senescence, you have a reduced root respiration under both low nitrogen and control conditions. And when you have root cortical senescence, you have a greater shoot dry biomass under low nitrogen conditions. And that's really driven by a reduced root respiration and this reallocation of nutrients from senescing tissue to other root growth and more shoot growth. And that really drives greater plant growth and deeper rooting under low nitrogen. And just like MCS, root cortical senescence can have several benefits and trade-offs depending on the different environments. So when you have senescence of the cortex, you have reduced root respiration, like I mentioned earlier. However, we've also shown through tracing studies that you have a reduced radial transport of nutrients, and you also have a reduced radial hydraulic conductivity, which can have benefits or negative effects depending on the environment. Also, of course, when you have senescence of the cortex, you don't have a living cortical habitat for AMF colonization and other things. So there's potential benefits and trade-offs in specific environments. Now I want to put those two traits together, and this is really what we're interested in and focusing on now, is how the root cortex can develop multiple tissues simultaneously or successively. So this is just an example. So early in growth, you have simple cortical parenchyma, these thin-walled living cells. In that same root cross-section a little later in growth, you might develop multiserrate cortical sclerenchyma, this lignin in the outer cortical cells that's marked with a red arrow. A little later in growth, you might have also root cortical senescence formation starting, marked with these yellow arrows. And the root cortical senescence formation can continue to progress. Eventually, the entire cortex will senesce, only leaving the stele. So that is an example of successive and simultaneous tissue formation in, for example, wheat in the root cortex. Now we can actually see that in greenhouse and field studies. So this is a collaboration with CIMMYT. This is wheat root cross sections, and this is near the root tip here. This is near the root shoot base, so near the root shoot junction. And you can see there's different combinations of these tissues that can form. Some genotypes form a combination of MCS and RCS. So you can see at the root tip, you have relatively simple root cortical parenchyma, and then you develop MCS. Then you develop MCS and root cortical senescence that will eventually progress into complete root cortical senescence, which you see here, which a little remnant of maybe MCS or an epidermis, maybe because it has more lignin or superin, and it's just takes a little bit longer to degrade, and eventually only leaving the stele. We also have genotypes that just form MCS, which you see here. You have cortical parenchyma near the tip, and then you have MCS that persist throughout plant growth. And we also have genotypes on the bottom row, for example, that just form RCS. So again, you have cortical parenchyma near the root tip, and then you can see with this yellow arrow, root cortical senescence formation, eventually only leaving the stele. So roots can form, these plants can form these different combinations of these tissues together, or they can have these tissues sort of in isolation. And what we see in the field is that when you have these combinations of tissues, you have an advantage under drought. So in wheat plants, when you have a combination of MCS and RCS, you had 24% increase in yield under drought compared to just genotypes that had cortical parenchyma that persisted throughout plant growth, or genotypes that had MCS in isolation or RCS in isolation. So if you just form RCS, we see no yield benefit under drought. If you just form MCS, we see no yield benefit under drought. When we have a combination of these two, that's when we really see this yield benefit. And the reasoning for this is because we are hypothesizing, right? We have no experimental evidence for this, but near the root tip, you have an intact cortex, also some MCS developing, where you can penetrate hard, dry soil, and then also take up radially water and nutrients. And then in these older tissues, you have root cortical senescence developing that result in sort of metabolically efficient soil exploration. So you have a reduced root respiration. You can remobilize nutrients from senescing tissue for further root growth, further shoot growth. And this tissue really doesn't radially take up nutrients and water anymore. In these older tissues, it just forms as an axial conduit, an axial tube to transport water from the root to the shoot. So near the root tip, you're penetrating, you're elongating into these hard, dry soils, you're radially taking up the nutrients and water. And in the older parts of the root, you just have this metabolically efficient soil exploration. So Larry York did a study several years ago showing that interactions between different traits can be additive, synergistic, or antagonistic. And here I think really what we're seeing with MCS and RCS is a synergistic combination, right? Because RCS in isolation, no yield benefit under drought. MCS in isolation, no yield benefit under drought. When you have them together, you get a 24%, a substantial increase in yield under drought in wheat. And Stephanie Klein also looked at these sort of integrated strategies of combinations of traits. So we grew a large maize association panel and phenotyped them for root anatomy. And this is what sort of the average root looked like here. And then we discovered that roots can have three main strategies that all can achieve drought tolerance. The first one can be reduced metabolic maintenance costs in the cortex. You can have parenchyma or root cortical senescence develop, large cortical cells, fewer cortical cell layers that really reduce the carbon and nutrient costs of construction and maintenance of the root tissue. A second strategy could be restricted uptake of water to conserve soil moisture, so you can have many small xylem vessels that reduce axial hydraulic conductance to promote water banking, so you can moderate shoot water stress throughout the growth season. You can keep water in the soil to keep your root tips hydrated to keep growing. You keep enough water in the soil, so at the grain filling stage or at the reproductive stages, you have enough soil moisture to make that happen. And the third strategy might be improved penetrability of dry, hard soil. So you have a large diameter root with a large stele and MCS in the outer cortical cells that can really help the root elongate and punch through that hard, dry soils. And through the information, the data I've shown today, maybe there can be a fourth strategy, fifth strategy. I think the number of combinations to achieve some stress tolerance there's a lot, right? So I propose here strategy 4, which is a combination of strategy 3 and 1. So near the root tip, you have strategy 3. You can improve the penetrability of hard, dry soils and take up nutrients. And then near the root-shoot junction in some of these older root areas, you can have metabolically efficient soil exploration when you have a combination of both root cortical senescence and multiserrate cortical sclerenchyma. I want to emphasize that this is very, very complicated phenotypic space that we're still trying to dig into. So we know that there's significant variation for the combinations of these traits that can form in the cortex, and the timing of this tissue formation is also really important. So for example, here I give just an example of one genotype under drought stress. You can see, you've seen this before, MCS progressing into RCS. But under that same genotype under phosphorus stress, root cortical senescence progresses much faster and it occurs much closer to the root tip. And in that same genotype under nitrogen stress, you have root cortical parenchyma sort of thrown into the mix. So parenchyma have parenchyma tissue form, and then MCS form, and then that parenchyma sort of turns into root cortical senescence later in growth. So in summary, root anatomical and architectural traits have important roles for water capture and stress tolerance. So I described root cortical senescence, multiserrate cortical sclerenchyma. I touched on parenchyma, but there are also many, many others. Root traits interact in time and space, and deciding which root is representative, which root class, which nodal origin of the root and under which developmental stages and at which time points is really, really critical, but a really, really difficult question to answer. And there are many different integrated phenotypes that can achieve the same goal for stress tolerance. So there's not one optimal traits. There's many, many combinations of root traits that it can achieve drought tolerance, nitrogen stress tolerance, and other stress tolerances. So that's all I have. I really want to acknowledge my group at IPK, especially Yvonne Lopez Valdivia, who helped with the root collection and sampling work of the wheat plants in Simit in Mexico, and Dylan Jones, who really led the anatomical imaging device. The root cortical senescence work was done at the Forschungszentrum Jülich. The multiserrate cortical sclerenchyma work was done at Penn State University in collaboration with Jonathan Lynch. Thank you for your attention.